Welcome back to Weekly Neurology Deep Dive, the show that keeps you up to date with the latest in neurological research, curated specifically for busy neurologists.
That's right. We're going to break down some key takeaways for your clinical practice.
And don't forget, this podcast is for educational purposes only, no commercial affiliations.
Think of this as a highlight reel to pique your interest and encourage you to read the full paper for a deeper dive.
This week, we're taking a deep dive into Friedreich Ataxia.
FRDA for short.
Let's travel back in time to the 1860s, when Nicholas Friedreich first described this condition.
Initially, he attributed the spinal lesions he observed to inflammation.
Imagine that.
It really highlights how medical understanding evolves over time.
It's fascinating how his observations, although incomplete, laid the foundation for our current understanding.
Right. And fast forward to the 1970s.
We see the impact of patient advocacy in driving research.
Claude St. Jean, diagnosed as a teenager, became a powerful advocate urging neurologists to study the disease.
And his determination led to the creation of the first FRDA Patient Association.
And the launch of the Quebec Cooperative Group on FRDA.
A pivotal moment in the history of this condition.
Alongside Dr. Anita Harding's research in London, the group's work led to much-needed diagnostic criteria for FRDA.
And these criteria were essential for large-scale genetic studies,
which ultimately revolutionized our understanding of this condition.
The late 1980s and early 1990s witnessed an explosion of research.
Using advanced genetic techniques like linkage analysis.
Restriction fragment length polymorphisms, or RFLPs.
Exactly. Powerful tools for pinpointing gene locations.
Researchers narrowed down the location of the FRDA gene to chromosome 9.
A collaborative effort, with researchers across the globe joining forces.
And within this region, they found a specific transcript later named X25.
Initially seemed promising, but when they looked for mutations in X25,
they only found them in about 2% of patients.
A puzzling finding, but researchers noticed that X25 cDNA levels were lower in patients compared to controls,
hinting at something silencing the gene's expression.
The gene was there, but something was preventing it from doing its job.
Further investigation revealed the presence of extra DNA in patients and carriers,
leading to the discovery of a GAA repeat expansion within an ALU sequence.
They had found the culprit behind FRDA.
And this breakthrough paved the way for PCR-based diagnostic tests,
changing the landscape of FRDA diagnosis.
Now, let's talk about this GAA repeat expansion.
It's fascinating that it's found within an ALU sequence,
a type of repetitive DNA unique to humans.
And it's these expanded GAA repeats, what we call E alleles, that are the primary culprits behind FRDA.
Found in populations with European, North African, Middle Eastern, and Indian ancestry.
Present in over 1% of individuals.
Now, there are also normal GAA repeats with around 8 or 9 units.
We call those SN repeats.
And longer, but still non-pathogenic, LN alleles.
Found worldwide.
The key difference lies in the instability of the E alleles.
They're prone to expansions, especially during maternal transmission.
This instability leads to variations in the length of E alleles,
directly impacting disease severity.
Even at a somatic level, repeat sizes vary across different tissues within the same individual.
So even though someone inherits these E alleles,
their impact can be modified by these expansions?
Precisely. This instability also presents challenges for creating accurate animal models.
Research suggests that FRDA likely originated from a single event during early human migrations out of Africa.
The prevalence of FRDA in certain European regions might be linked to the persistence of E alleles in Ice Age shelters in northern Spain.
Fascinating, isn't it?
It's incredible how these ancient migration patterns may have shaped the genetic landscape of FRDA.
So we have this GAA repeat expansion impacting the FXN gene.
What exactly does this gene do?
The FXN gene is responsible for producing a protein called frataxin.
And this protein plays a critical role in assembling iron-sulfur clusters,
or ISCs for short.
Essential cofactors for various enzymes involved in vital cellular processes like respiration, metabolism, and antioxidant defense.
Without enough frataxin, these processes are significantly disrupted.
So the GAA repeat expansion leads to a deficiency in frataxin,
which in turn disrupts ISC production,
triggering a cascade of issues: mitochondrial dysfunction, oxidative stress,
ultimately cell death.
And what's even more concerning,
certain cell types are particularly vulnerable in FRDA.
Like
neurons in the cerebellum, spinal cord, and peripheral nerves are especially sensitive,
as are cardiomyocytes in the heart.
This explains the characteristic symptoms of FRDA,
like ataxia,
problems with coordination and balance,
as well as cardiac complications.
So a single genetic defect can set off a chain reaction with far-reaching consequences.
And to complicate matters further, The body's attempts to compensate for this frataxin deficiency often backfire.
exacerbating the problem.
How so?
When cells sense a shortage of ISCs, they try to increase iron uptake.
Seems logical since iron is a key component of ISCs.
But the issue isn't a lack of iron, but rather the inability to properly utilize it due to the frataxin deficiency.
So this increased iron uptake ends up overloading the mitochondria with iron.
Leading to further oxidative stress and cellular damage.
It's like the body's trying to put out a fire with gasoline.
A great analogy.
As research progresses, we're starting to understand that FRDA is even more intricate than we initially thought.
We're starting to see a much bigger picture, with frataxin deficiency impacting a complex network of pathways and processes.
And this deeper understanding has been critical for developing more targeted and effective therapies for FRDA.
Which we'll delve into next time. Welcome back to Weekly Neurology Deep Dive. We're picking up our conversation about Friedreich Ataxia, focusing on what you, as neurologists, need to know to apply these findings in your practice.
Last time we journeyed through the history of FRDA, highlighting the pivotal role of patient advocacy.
And the groundbreaking discovery of the GAA repeat expansion in the FXN gene.
We also explored the cascade of consequences stemming from frataxin deficiency,
Including disrupted iron-sulfur cluster production, mitochondrial dysfunction,
and the unfortunate tendency for the body's compensatory mechanisms to worsen the situation.
Right.
And we touched upon how this deeper understanding of FRDA has paved the way for more targeted therapeutic approaches.
Which is where we'll focus our attention today.
So,
While early attempts to address oxidative stress and iron abnormalities with general antioxidants and iron chelators showed initial promise,
they ultimately fell short in clinical trials.
the focus has now shifted to therapies that precisely target the specific pathways disrupted by frataxin deficiency.
One of the most notable success stories is omaveloxolone, a drug that activates NRF2,
A key regulator of the body's natural antioxidant defenses.
a pathway we know is compromised in FRDA.
And this isn't just a theoretical success either,
Omaveloxolone has shown significant clinical benefits.
leading to improvements in neurological function for many patients.
And significantly.
Omaveloxolone has received approval from both the FDA and the European Medicines Agency.
Making it a real game changer in the treatment of FRDA.
This approval marks a major victory for the FRDA community.
And offers hope for a brighter future for individuals living with this condition.
So,
We have a real tangible treatment option now.
what other promising drugs are in the pipeline?
Vatiquinone is another potential treatment.
That specifically targets the 15-little pathway, which is involved in oxidative stress and cell death.
While an initial randomized controlled trial didn't show a significant difference in ataxia scores compared to a placebo.
An open-label extension study has yielded encouraging results.
Based on these findings,
A new drug application has been submitted to the FDA. If approved, vatiquinone would provide another valuable tool in our arsenal against FRDA.
that would be fantastic, offering patients more choices and hope.
Absolutely.
Are there other therapies on the horizon targeting different aspects of the disease?
Absolutely. Liraglutide,
A drug that targets the PGC1 alpha pathway involved in mitochondrial biogenesis and function.
has also shown positive signals in early clinical trials.
While the primary outcome of the trial,
A measure of cervical spinal cord volume, liraglutide did demonstrate encouraging improvements in secondary outcomes, like changes in cervical spinal cord metabolites measured by MRFs.
and enhancements in upper limb dexterity.
This suggests that liraglutide may still hold therapeutic potential.
So it's not a one-size-fits-all approach.
We're seeing a diverse range of therapies targeting different pathways affected by frataxin deficiency.
It's truly remarkable how our understanding of the disease is shaping the development of these targeted therapies.
And this progress wouldn't be possible without the invaluable research models that have been instrumental in advancing our understanding of FRDA.
And facilitating the development of these novel therapies.
These models range from simple organisms like yeast to more complex animals like mice.
And they've played a crucial role in unraveling the complexities of FRDA.
And evaluating the efficacy of potential treatments.
Yeast models, for instance, have been instrumental in understanding the role of frataxin in iron-sulfur cluster biogenesis.
A key process that's disrupted in FRDA.
These simple yet powerful models have provided insights into the consequences of frataxin deficiency,
Such as mitochondrial iron accumulation and dysfunction.
mirroring some of the key features observed in humans.
Yeast models have also been essential for initial drug screening.
And identifying potential therapeutic targets.
They've laid the foundation for more complex models.
Right.
Fruit flies, or Drosophila, and the nematode worm C. elegans.
Have also contributed significantly to FRDA research.
These models, with their more complex nervous systems, have allowed us to investigate the neurological consequences of frataxin deficiency.
We can study how the lack of frataxin affects movement, coordination,
And other neurological functions.
but I imagine that the most extensively studied models are mice.
Given their closer resemblance to humans.
Yeah, absolutely right.
Mice are mammals, like us.
And their genetic makeup is remarkably similar to ours.
This makes them excellent models for studying human diseases.
However, replicating the GAA repeat expansion in mice with its inherent instability
Has posed a significant challenge.
and the fact that FRDA affects multiple organ systems adds another layer of complexity,
Despite these hurdles, researchers have developed a diverse array of mouse models
each with its strengths and limitations.
that have collectively advanced our understanding of FRDA.
For instance, we have conditional knockout mice.
Or CKO mice, where the FXN gene is deleted in specific tissues or cell types.
These models allow us to study the precise effects of frataxin deficiency in a targeted manner.
The EKO-MCK model, for example, where FXN is deleted in the heart and skeletal muscle, accurately reflects the cardiomyopathy seen in FRDA.
And the EKOP valve model
With FXN deletion in proprioceptive and cerebellar neurons.
mimics the sensory and cerebellar ataxia characteristic of the disease.
It's like having different lenses to examine the multifaceted nature of FRDA.
And then there are the mouse models that incorporate the GAA repeat expansion itself.
Yes, although replicating the behavior of these unstable repeats in mice has been a major hurdle.
The Kiko mouse, for example, carries a 230 GAA repeat expansion.
And a deletion of exon 4 in the FXN gene.
It develops neurobehavioral deficits
Glucose intolerance.
and cardiac dysfunction.
Providing valuable insights into the consequences of frataxin deficiency.
It's incredible how researchers have overcome these challenges to create models that mimic the human disease so closely.
And it doesn't stop there.
Humanized yeast artificial chromosome.
Or YAC models have also been developed.
The YG8R and YG8-800 mice, for instance.
Carry large GAA repeat expansions within the human FXN gene and exhibit somatic and intergenerational repeat instability, mirroring the genetic dynamics observed in humans more accurately.
So we have a spectrum of mouse models, each offering unique perspectives on FRDA.
But what about cell-based models?
How do they contribute to the research effort?
Cell-based models are indispensable for studying the cellular and molecular mechanisms of FRDA in a controlled and accessible environment.
Early studies relied on readily available cells from patients like skin fibroblasts
And peripheral blood mononuclear cells.
or PBMCs.
While these cells have reduced frataxin levels, they can still function relatively normally.
But they've been helpful for studying the basic biochemistry of frataxin deficiency.
And testing potential drugs.
Exactly.
However, to study the cell types directly affected by FRDA, like neurons and cardiomyocytes,
You need something more specialized.
that's where induced pluripotent stem cells,
Or iPSCs, come into play.
iPSCs, are those incredible cells that can be generated from adult cells and then reprogrammed to become virtually any cell type in the body, right?
That's right.
We can take skin cells from a patient with FRDA, reprogram them into iPSCs,
And then differentiate those iPSCs into neurons or cardiomyocytes, the very cells affected by the disease,
This allows us to study the disease process in a dish.
providing a level of detail and control that wouldn't be possible with animal models.
What kind of insights have iPSC-derived models provided?
iPSC-derived cardiomyocytes from FRDA patients, for instance, display clear mitochondrial abnormalities and impaired function.
Consistent with the cardiac complications observed in the disease.
And neurons derived from FRDA iPSCs
While subtly different from those of healthy individuals,
have revealed delayed electrophysiological maturation
reduced levels of iron-sulfur cluster proteins,
and signs of oxidative stress, mirroring the key cellular features of the disease.
So these models are accurately recapitulating the key cellular features of FRDA.
Providing a powerful tool for studying the disease's underlying mechanisms.
Precisely.
And the advancements don't stop there.
Researchers have even generated iPSC-derived sensory neurons.
Particularly proprioceptive neurons.
The very neurons that are exceptionally vulnerable in FRDA and responsible for our sense of body position and movement.
These iPSC-derived sensory neurons have shown altered neurite extension.
Reduced expression of proprioceptive markers.
And even slightly impaired excitability, mimicking the sensory impairments seen in FRDA patients.
These findings suggest a potential developmental component to the sensory neuropathy in FRDA.
Indicating that these neurons might be compromised from the very beginning.
That's a significant finding.
And it highlights the power of these models to unravel the intricate details of the disease process.
Have iPSC-derived models been used to study other cell types relevant to FRDA, like glial cells?
Yes, they have.
Glial cells, those crucial support cells for neurons,
Are gaining increasing attention for their potential role in FRDA pathology.
studies using iPSC-derived microglia, the brain's immune cells,
Suggests that these cells might be intrinsically activated in FRDA.
potentially contributing to neuroinflammation and neurodegeneration,
This underscores the complexity of FRDA.
and the importance of understanding the interplay between different cell types in the disease process.
So it's not just about the neurons themselves, but also the intricate network of supporting cells that surround and interact with them.
It's truly remarkable how far we've come in developing sophisticated and diverse model systems for studying FRDA.
From simple yeast to complex human iPSC-derived neurons.
These research tools have been instrumental in advancing our understanding of the disease and paving the way for the development of new therapies. And as these models continue to evolve and improve,
Their role in the quest for effective treatments and ultimately a cure for FRDA will only grow.
the progress we've witnessed over the past 3 decades has been nothing short of extraordinary.
From a limited understanding of FRDA's genetic basis to unraveling complex molecular pathways,
Identifying potential biomarkers,
developing targeted therapies.
and exploring gene therapy and protein replacement approaches.
The advancements have been remarkable.
And it's been driven by the combined efforts of dedicated researchers, clinicians, patients.
And their families working together.
Absolutely. The FRDA community is a testament to the power of collaboration and the pursuit of knowledge and hope.
Initiatives like the Friedreich Ataxia Global Clinical Consortium
Or unify.
bring together researchers and clinicians from around the globe to conduct large-scale natural history studies and clinical trials.
Further demonstrating this global collaborative spirit.
Unify has been invaluable in furthering our understanding of how the disease progresses and evaluating the effectiveness of new treatments.
And we can't forget the crucial role of patient advocacy groups like the Friedreich's Ataxia Research Alliance,
Or FAR.
which have tirelessly advocated for research, raised awareness, and supported those living with FRDA and their families.
These organizations have played a pivotal role in driving the progress we've seen.
And continue to inspire the research community.
The story of FRDA research truly embodies human ingenuity, perseverance, and an unyielding belief in the possibility of a cure.
While challenges remain.
The progress we've witnessed over the past 3 decades is undeniable.
And the future of FRDA research is filled with promise.
Welcome back to Weekly Neurology Deep Dive. We're concluding our exploration of Friedreich ataxia, focusing on the latest advancements that are shaping the future of this field. In our previous discussions, we've covered the remarkable journey of FRDA research, from the initial descriptions to the discovery of the FXN gene, the development of targeted therapies, and the crucial role of research models in driving progress. Now, let's delve into a critical aspect of FRDA research that translates these scientific discoveries into tangible benefits for patients: clinical trials.
Clinical trials are the bridge between promising laboratory findings and real-world applications. They are the rigorous and carefully controlled studies that determine whether a new treatment is both safe and effective for humans.
Conducting clinical trials for rare diseases like FRDA presents unique challenges, doesn't it? The smaller patient populations and the variability in disease progression can make it difficult to recruit participants and interpret results.
That's right. However, the FRDA community has been a pioneer in developing innovative clinical trial designs and outcome measures tailored to the specific needs of this rare disease.
Can you give me some examples of these innovative approaches?
One notable example is the use of natural history studies, like those conducted by the Friedreich Ataxia Global Clinical Consortium, or UNIFY. These studies provide a baseline understanding of how the disease naturally progresses and help identify sensitive outcome measures, which are crucial for interpreting clinical trial data.
So these studies are essential for determining whether a treatment is genuinely slowing down disease progression or simply masking symptoms. What other innovations have been implemented in FRDA clinical trials?
The FRDA community has been proactive in developing standardized clinical rating scales like the Friedreich Ataxia Rating Scale, or FARS, and the Modified Friedreich Ataxia Rating Scale, or mFARS. These scales provide a more objective and quantitative way to assess disease severity and track changes over time, making it possible to compare results across different studies and patient populations.
That makes sense. Having standardized tools is essential for ensuring consistency and reliability in clinical trials. But recruiting enough participants for clinical trials, especially for a rare disease like FRDA, must be incredibly challenging.
It is, however, researchers are employing several strategies to address this. One approach is to conduct multi-center trials involving multiple research sites across various countries and regions, casting a wider net to reach more potential participants. Organizations like Unify play a crucial role in facilitating this global collaborative approach.
So expanding the geographical reach of trials is key. Are there other ways to make clinical trials more accessible to individuals with FRDA?
Another strategy is to develop more inclusive clinical trial designs that allow for a wider range of patients to participate, even those with more advanced disease or certain pre-existing conditions. This expansion of eligibility criteria is particularly important for FRDA, where disease progression and severity can differ significantly from person to person.
That's an important consideration, ensuring that trials are representative of the diverse patient population. Now, what about the ethical considerations of using placebos in clinical trials? Denying some patients a potentially beneficial treatment, especially in a rare disease with no known cure, seems ethically challenging.
You raise a valid point. The FRDA community has been at the forefront of advocating for alternative trial designs that minimize the use of placebos while maintaining scientific rigor. One such approach is the delayed start design, where all participants eventually receive the treatment, but some begin later than others. This allows researchers to compare disease progression in those receiving the treatment early versus those who start later, ensuring that everyone benefits from the treatment while still providing valuable data on its effectiveness.
That's a great example of how ethical considerations are shaping clinical trial designs for rare diseases. It's inspiring to see the FRDA community actively tackling these challenges and developing creative solutions. This commitment to both scientific rigor and ethical considerations is truly commendable.
I agree, and this dedication is evident in the remarkable progress we've witnessed in FRDA research over the past 3 decades. From unraveling the genetic basis of the disease to developing targeted therapies and implementing innovative clinical trial designs, the FRDA community's unwavering commitment to advancing knowledge and improving the lives of those affected by this condition is truly inspiring.
While challenges remain, the progress made so far instills hope and fuels the determination to continue pushing the boundaries of research and strive for a future where FRDA is no longer a life-limiting disorder. The collaborative spirit of the FRDA community, the innovative approaches to clinical trials, and the dedication to both scientific progress and ethical considerations offer a beacon of hope for a brighter future for those living with FRDA.
That's a sentiment I wholeheartedly echo. As we conclude this deep dive into Friedreich ataxia, we encourage you, our listeners, to delve further into the research, connect with the vibrant FRDA community, and stay informed about the exciting advancements on the horizon. The journey is far from over, but the future holds immense promise.
That brings us to the end of our three-part exploration of Friedreich ataxia. Thank you for joining us on this journey. Until next time, stay curious, stay engaged, and continue exploring the fascinating world of neurology.
Goodbye, and thank you for listening.